Hello, my name is Joseph Tagabon. I'm 16 years old, a cord blood recipient. I was first diagnosed with AML in January of 2021 and relapsed in July of 2022. I had my bone marrow transplant in December 22nd, 2022. I was fortunate that our bone marrow team found a match. Without cord blood donors, I wouldn't have had a second chance at life. I'm back home in Petersburg, Alaska, after being away for so long. I'm enrolled in high school, currently trying to catch up my sophomore year that I missed the entire year. At the same time, studying uh, junior subjects. It's a great feeling to step into my school ground and see familiar faces of friends and teachers. I even started shooting basketball as a way of my side passion. Although it's hard because of my physical condition, um, a positive attitude helps me to push myself to stay better. I am hoping to be better and to join in-person class after my 1 year post-transplant in summer. I'd like to personally thank the bone marrow transplant team and the Fred Hutch and Seattle Children's Hospital. I'm fortunate enough to receive a cord blood bone marrow transplant, and it saved my life from a horrendous disease. Without any expertise, I wouldn't be in front of you all right now. I owe my life to my bone marrow transplant team and my donor.
Hello, my name is G, and I'm Joseph's mom. As a parent, I'd like to encourage mothers to consider banking cord blood. It saves lives, and my son is a living testimony. There are those that are still waiting for a donor, and you may be the match. To the mother of my son's donor, I'd love to tell you this. You have done your purpose in life. You are a lifesaver. God bless you and yours. Thank you.
Good morning, everyone. A couple of things first. One, that was like not a fake background for him. That he's actually only about 4 months post-transplant, and he looks fantastic. And he's from St. Petersburg, Alaska. So, I mean, he's really from a very remote region. So, I think we are grateful to everyone here that we can help treat patients like Joe. So, okay, another thing is, I'm going to make some plugs. Please follow us, the Cord Blood Association, on LinkedIn and Twitter, if you know how to do that. I don't know how to do that, but tag us and give us some shout outs if you've been here at the conference. Okay, so with that, I'm trying to think, do I have any other housekeeping things? I don't think so. We can start this last session, or second to last session. Thanks for sticking it out and staying till the end. Okay, do I need to advance this, or do you guys? Let's see. Okay. Okay, so this is a session on utilizing cord blood and perinatal tissues as starting materials for cellular therapy. I am the moderator. These are my disclosures, really with my roles at 2 different companies, one where I hold equity and am an inventor on patents. Okay, so the speakers. We have an excellent group of speakers here today. And really, we hope to facilitate discussion. That's really the goal of this. These speakers have a wealth of knowledge in this space of cell and gene therapy product development. 2 of the speakers are representing companies that utilize cord blood as starting material, and then we have a third speaker, who is from a company that is generating scalable human MSCs and exosome product systems for end users, so research use only and clinical grade products. So Jesse Katari is the CSO and co-founder at ImmuneBridge. They are working on development of cord blood-derived NK cells. And then we have Tara Sadeghi, who is the Chief Operating Officer at Cellenkos. Tara has a wealth of experience and has worn many, many hats. I think too many hats to even talk about here, all the way from, I think, really research, clinical ops, regulatory and quality, so a wealth of knowledge there. And then we have Joe Takas, did I say it right? Who is a bioprocessing application scientist at RoosterBio, also is going to bring sort of that side of things, the non-therapeutic side of things, but utilizing cord tissue. The session outline, again, this is meant to be a very focused discussion with people from industry and really speaking to the people in the audience, the cord blood bankers, the end users for some of these therapeutics, to really have a discussion around why cord blood and hopefully what we'll tell you is what we need as a sponsor from the cord blood banks, and what are we going to do and how are we going to account for all of the success that I feel very confident we will have in the future with these products. So the outline is that we'll have some brief speaker presentations first, probably about 30 to 40 minutes in the beginning, and then we'll do a moderated panel discussion that I will lead, followed by an open Q&A. So please be thinking about your questions ahead of time. So I think I showed this slide last year as well, but this slide just gives me a lot of faith and hope and excitement in the space of allogeneic cell therapies. And it really demonstrates that there has been a huge and steady growth over time in the cell and gene therapy landscape. This is specifically looking at oncology indications. And what you can see here is that while there are a number of therapies underdeveloped, relatively few have been approved. But even with that, it's a very active pipeline. And there's been a big increase even just between 2020 and 2021. And then if we look at auto versus allo, and we're really going to focus here on allogeneic cell therapies for this session today, even though autologous cell therapies they still outnumber allogeneic cell therapies for many reasons that I think we've all discussed over the last 2 days. However, if you look at the allogeneic growth, the number of products that are in preclinical or phase 1 are increasing tremendously year over year, as are the number of allogeneic products in phase 2 development. And I think we're going to be pretty close to seeing some of these products. And shout out to Gamida for getting an allogeneic cell therapy FDA approved. So the panel up here is really, again, focused on allo, but not patient-specific necessarily. And then this is, a plug for the CBA. We really value our members. We are working hard to understand what can we do as an association to provide value to all of you sitting here in the audience, and to really be that bridge and that conduit for collaboration and networking. And you can see here's a membership map for the association, and we would love, 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 love to add more of you there. With more members, we will all have more voice and be more active in the space. Okay, so with that, I'm going to invite, I guess Jesse. It was going to be Joe, but Jesse, you can come up first. We'll go with the order of the slides, and he'll give his presentation. Thanks.
All right, good morning, everyone. So when I initially wrote this talk, I was thinking really about our technology and really about how deep screening and donor variability and the expansion technology. But over the course of having a bunch of conversations over this weekend, realized that what we're here to talk about is the fact that this is new partnership possibilities that will allow us to work closely with cord blood banks to really find ways to get this supply that we need to do the most for the most patients. So this is not a patient. This is actually my son, Huxley S. Kotari, who, if you're keeping score, his initials are HSC, which I didn't realize until I was writing it on the birth certificate form, but a little happy twist of fate. But we wanted to bank his cord blood, and actually because we know ability of cord blood to be a cure for people, we wanted to donate it to a public bank. But it turns out even in places like San Francisco, at UCSF, it's difficult to bank cord blood. But we were able to do it privately. And what the great part about that is that we actually got to see some statistics on what would have been in this cord blood, which, as you realize, if you look at those numbers, they wouldn't have made it into a public bank because clearly they fall below the threshold. Sorry, buddy. Yeah. But we see the value differently, and not just because this is my kid, but because as a company that makes therapies from cord blood units, we actually have technology that can allow us to do something with this. So because we have a model that is different for sourcing and screening CBUs, we can take this, turn it into valuable therapies, and what we need is a way to interact with the cord blood banking industry to make that possible. So, at the core, what started ImmuneBridge was technology that we have that allows us to expand CD34 cells from cord blood or other tissues, but cord blood is far and away a better expander. And we've done this starting from purified CD34 cells. We've done it starting from segments. We've done it with full CBUs. And the data on the left is the early data. That's 7 different donors, vials of cells purchased, purified. On the right is 14 different segments run in parallel. Those are the dots. And the lines are the full units run through a bioreactor. So we're able to do this at very small scale and very large scale. And at the end, we have very multipotent cells. So the cells we've taken through down various differentiation methods to everything from macrophages to NK cells and demonstrated functionality of those cells. What we are focusing on is natural killer cells. And natural killer cells are naturally potent cancer killers. You don't have to give them a CAR for them to know what to do. And the reason that we're starting from expanded cord blood CD34 cells is because, one, it allows us to screen CBUs from segments. It enables us to do large-scale production where we can make thousands of doses from a single cord. And one of the other things that we're coming to see is this expansion allows us to treat diversity and variability as a strength. So taking HSC variability and saying, well, actually, that allows us to pick the units that will make manufacturing better. Natural killer cell variability allows us to say, well, these are going to be the therapies that will work better. So natural killer cells, to dive in for just a minute, and you've had a lot of priming this weekend, so I'm not going to go too deep. I'm actually going to go in the direction to say why they are very variable. So something you know is that HLA and KIR genes are the most diverse loci in the genome. But those aren't the only ways in which NK cells are able to interact with cancer and decide what to kill. In fact, they have dozens of receptors that tell them where to go, whether to live or die, and whether to kill. And we have direct data, even in our hands, showing, yes, in fact, the NK cells that come from different donors are incredibly variable. You can have killing that ranges from 90% to 20%, and this is reproducible. Each of these is a paired donor that was run from 2 different segments that were from the same cord but created in isolation. So it's very reproducible on a per donor basis, but from donor to donor, it's highly variable. So we had to ask the question of, So then there's the question of, well, can't you just engineer your way out of this variability? Can't you just put a CAR in the cells and get rid of the fact that they are so variable? And so there's a paper that we were looking at a while back where you see, yeah, in fact, you can take NK cells and you can make them more potent by putting a CAR in them. And I was very excited about this paper, and we started digging into it deeply, looking at the axes, realizing, hold on a second. if you rescale this so that you're actually looking at them on the same axis, you can see that this non-CAR line on the left is actually more potent than taking a CAR and putting it into a donor that had weaker NK cells. So choosing the right donor can actually be more of a boost than just adding a CAR. Of course, then we can engineer a CAR in on top of that. But the first goal here is to find the best source. So how do we efficiently do that? Well, usually when I'm showing this slide, it's because the people that I'm talking to have never seen a CBU before, and certainly not in real life. And just, since we want this to be very interactive, how many of you in this room have actually seen one of these in real life? Like, show of hands here. Yeah, I love that. And then how many people have actually held one in real life? Wow, okay. I didn't expect it to be that high, but this is even better. So you understand like the true scale of these and how tiny actually some of the starting material can be, how small a segment is. But because we have the expansion technology, we can run very meaningful assays, so full panels of 14 different killing assays, expansion data, even to the point where we have enough to run mouse experiments starting from just a segment. And when doing the math, we realize we can essentially turn a segment into a CBU's worth of CD34 cells. But then the great thing is, at the end of that, we still have the full CBU left over. So we can go back to that actual CBU that we took, we ran through all of those assays, and put it into a bioreactor so that we have material for clinic. And that's what we're here to talk about, is how to enable this model. We say screen the segment, translate the cord, and that's what we are looking to do. So the screening model looks like this. So step one, acquire lots of segments and expand them, run them through assays, take the ones that score well in those initial high throughput assays, run them through more intensive assays, and that's things like patient-derived tumoroids, mouse models, that allow us to really say which are the best therapeutic cells, and then take those CBUs and expand them out for clinic. And what we need here is 100 microliters of processed cord blood. So we use segments. It can be flexibly done in other ways. So the piece that we also see as enabling this is the ability to look at the genetics of the NK cells, so to identify the CBUs ahead of time that can improve NK cell function. And we have the empirical screening that is core to our technology. That is fully enabled by the ability to expand the HSCs. The genetic screening is something where we can rely on both published data already. We can also create our own data as we go through and we learn what the signatures are of better NK cells. So when looking for ingredients to this, what do we need? Well, first of all, we need translatable CBUs. And this is something we've had a few conversations with over the weekend of what does it mean for a CBU to be good for an immunotherapy, but not necessarily at the level that you would use for a transplantable unit. And this is something that for us means a much lower TNC. So we can start with 1 million CD34 cells. So that's about 2E8, about 2E8 TNCs. And this is something that we're here to say we want to collaborate with the cord blood banking industry to just create a common language around and to define this category. The other thing that we need in here is access to genetic information. So a lot of you have blood that has been spotted on filter paper as part of the collection process. So things that we can go to and sequence a handful of genes. And a handful is less than 100, probably 20. And from this, that allows us to say which ones are we going to take, which segments do we want to take through to screening, And that's the third component we need, is some amount of material that we can screen first. So this is something that many cord blood units already have. This is something that is, you're using it, and we want to use it in a different way. Okay, so practically speaking, yes, this is a good slide to take a picture of, especially if you're a cord blood bank, because this is the thing that we are looking to do first, which is take the cord blood units that are already in the bank and analyze them to identify the ones that are most useful in this model, right? So this is, we'll talk about what that is in the next slide. Run the genetic screening, match it against a profile, and if it matches that profile, then send the segments for empirical screening at ImmuneBridge, where we rank it. And then at the end, once we've gone through this process of doing the empirical screening, we want to then take those units out of your inventory and send them to ImmuneBridge. And there's sort of a few metaphors for this, right? And I think it's like tasting the ice cream before you buy it, right? But what we want to do is think about how to include a partnership model that includes all of this in one. We don't just want to have a bunch of free samples before we buy something or not, right? That's not the flexibility that we're looking for. The flexibility we're looking for is a way to build a partnership that encompasses all of this from end to end. So, to paraphrase a folksy Marcy Finney from yesterday, nobody bakes a pie to have it thrown in the trash or sit in the freezer for 10 years. And so what we're talking about here is a way to expand the pie of units that are able to be used. So the first upper right quadrant, right, those are the high TNC rare units that are going to be somebody's only shot at a life. And we don't need those. We want those to go to those patients. And then a second tier below that are ones that have high TNC, but they're not as rare. And those also, I mean, they could be used, but they're not the ones that we think are really the sweet spot. For us, the ones that go into stage 1 are the translatable units, right? Lower TNC, below sort of the threshold that are likely to be used, but and the HLA can be really wherever it. We don't have requirements for that, essentially. So that's stage 1 of using what's already banked. But stage 2 is then saying, all right, how do we build this deeper relationship where we are actually thinking about what is going into the bank in the first place, and how do we make choices to expand the number of units that are available for this. So this is lower TNC threshold, and one of the things we've been talking about is how to include that genetic screening rapidly so that in the time that it takes before you've even run through the expense of processing unit, manufacturing it, using those consumables, that we're able to make a quick decision on whether or not that's something that is useful for immunotherapy. And that's something that we want to build out as part of stage 1. We want to understand what those criteria are so that you're able to know maybe it's useful for us, maybe it's not useful for us, but there is another criterion about that makes it more useful for other applications. Right, so that's really the full model. So the final thoughts. A lot of these are on practicalities, right? But we're here not just to talk logistics and practicalities, because I know that what gets a lot of us excited is the prospect of saving more lives from a single cord than would be possible otherwise. So we want to learn from screening. We want to do this with we have simple requirements. We don't, there's a more complicated model down the road, but initially it's pretty simple. And we really want to develop close relationships to enable a broader and deeper banking process so that we can take a single cord and decide whether that's going to save one person's life or whether that has the potential to be thousands of therapies to save many other lives, without having to make it a trolley car problem. So with that, we just want to say thank you. So from HSC and the HSCs, thanks for your attention, and I look forward to a great discussion. Ooh. Well, that's interesting. All right. Yeah. Oh, there we go.
Looks like it's Tara. So.
Good morning. I'd like to talk about a little bit about our company, and everyone is talking that how to build a relationship with cord blood banking and pharma, biotech. I want to talk about a little bit about we have started to build this relationship, and thank to the banks that they were patient with us, and I think there is a area that we can expand and build more. But a little bit about what we do in Cellenkos and with the cord blood. Cellenkos is a developing allogeneic cell therapy solution for treating rare inflammatory diseases. We have leveraged about $38 million financing to treat over 60, right now about 68 patients across different diseases. We completed 2 phase 1 studies, and recently we have started to do 2 more studies. The first 2 that we completed in last year, it was the bone marrow failure study that we did. It was a basically directed 3 out of 6 HLA match, fresh product. It was the first product that we had in Cellenkos. And one of the things I have to add here, this technology was licensed to Cellenkos from MD Anderson Cancer Center. And they had a study, the GVH, and that's the way this study, when we started to work at Cellenkos, we didn't have any previous history. So FDA wanted us to start with the fresh and HLA match. And later on, with the COVID came along, early 2020, we wanted to do allogeneic off-shelf cryo. And I have to add, on the first part of the study that we did that was a fresh, FDA allowed us to play with some of the leftover product from the phase 1 study that we had. And that's what they gave us a little bit opportunity to work how to cryopreserve these products and do some stability study. And we were very fortunate when COVID hit, we were already had this product in freezer. And with the collaboration with Johns Hopkins Hospital, we treated 2 patients. They were single compassionate use IND, and based on that finding that we had, we did a IND during COVID. And I think basically my background is in cell therapy and cord blood banking. This could not be done if it was not cord blood. Cord blood was cryoed, it was ready to use, and basically our group that we were working with them, and I want to thank them, New York Cord Blood, and also I have to say St. Louis Cord Blood Bank. Donna Reagan was amazing to help us to get our work done, and Cleveland Cord Blood Bank and Texas Cord Blood Bank, that's in the South Texas Blood and Tissue Center. So they helped us during the COVID to ship cord blood to us. we worked and we cryoed and we distributed. Both studies, the first one, it was MD Anderson Cancer Center, single site, but for the COVID, the phase 1 study, the CK082, we treated 45 patients. It was multi-center, and again, I have to say, this was cryo off-shelf, so we could ship it to sites. It was Johns Hopkins, Columbia University, Wake Forest, UNC, and Baylor College of Medicine. Including in those 2020, we did about 11 compassionate IND, 6 ALS patients, and 5 ARDS. And currently this year, we started two studies that we are working on. It's ALS and myelofibrosis. I have to say, my background, again, our team is oncology, but it's right now we are going to a different field, neurology, and we see, I don't have all data here to show you, but it's a really promising future for us. And also, Insight Pharmaceutical during 2020, they partner with us that they are having a licensing option for our product for CK0804 for myelofibrosis, treatment of the myelofibrosis. Cellenkos has a small footprint of the manufacturing, and basically we felt at the beginning when we got this product licensed from MD Anderson that for commercialization we needed control. And also being a small company, we don't have a lot of money. A lot of it was that how we could work and make this product manufacture, and if we are going to do any changes, it would be under our control. So we did our manufacturing is an ISO 7, and also we have a little lab that we do scale up and tech transfer. The technology is coming actually from MD Anderson Cancer Center. As I said, what we felt was basically that, manufacture the product. We don't store it at our site. We collaborate with Cryoport. It's a little bit expensive, but they are reliable. And what the speaker before me was talking about, that getting a cord and making sure that we can expand it, and we do multi-doses. And I think that's where we are heading with the cord blood. And this is one of the things that it was the mission for us, that we would provide this product for a lot of people. And the cost, we bring down the cost, and also we would give this product that they can just infuse it at community hospital, not just the major hospitals. And additionally, one of the things I have to say, that when we were working with the cord blood, one of the things was that it's clinically great. We can get this product, store it, and thaw it, and use it at the time we want it. And also, it's a uniform storing material. Already is being known, FDA has approved it. We work with the sites that they are FDA licensed, FACT, NetCourt FACT, or AABB accredited. And I have to, one of the other things, say that the cord blood banking is a lot of work. All of you all know that. And what we are fortunate to work with this cord blood banks, that they understand we are a small startup company. We don't have a lot of money. Future is going to change if this product is going to be commercialized, but they are working with us. They work with us that, when we get the product, they understand that we are working under IND. They understand that we are working in the regulatory framework that is required for their bank, and also we work with the groups that when they receive our product, that they are, so far all of the sites that we have worked with, they have a cell therapy lab, and we work in a way that we make sure that they are FACT accredited or AABB, and also our product is the way manufactured is acceptable within their standards. This is the pipeline so far we have. As I said, that we have completed 2 studies, phase 1 study, and 2 of them that basically right now we are enrolling. Both of them, they are multi-center. The ALS study is Columbia, UC Davis currently, and VA Hospital in Houston and Baylor. And for myelofibrosis is MD Anderson Cancer Center and UC Davis. We are working on Parkinson. And again, these are the diseases that there is no treatment for them. Cord blood, it is the future for us, and I think for many other companies. Overall, I wanted to just add, Cellenkos is a company that we work and we wanted to have an off-shelf product. And also, our product there is no HLA match. We are very fortunate. We have cords that they come with the segments. We are keeping the segments, and we are keeping some of the products for future that if we have to do any more research and some of the testing and all that. But again, I want to add that the banks that we work, it was a long process, but I think most of us right now that we work with this cord blood banks, they are willing to work with all the companies, I think. At least the ones that the 5 that I named, and there are few more that are being added to our list. And the reason for it is, I think, when we paired up with Insight Pharmaceutical, they are the company that they work with the small molecule. They have a first leukemia, Jakafi product. However, it's been so challenging to explain what's cord blood. There are cord blood that are available for future, and what is cell therapy. And there are companies that they like to be in this field, but everyone is afraid because they think it's very expensive. Yes, it can be expensive if they are coming from the pharmaceutical end, they build their GMP, and then after 2 years, they come and they do their manufacturing. I think we were fortunate in Cellenkos. We had a lot of experience. Majority of our staff, they are from MD Anderson, and also the technology was developed with the cord blood from MD Anderson Cord Blood Bank. Dr. Schwal was our vision that, how to work with this product, most of us. And the reason, again, we use cord blood, not just it's available, and you can just order it and get it, after everything is in place. However, it's better than the peripheral blood. We know that the Tregs in peripheral blood is flip-flop. It's not with cord blood. We know that. It's a stem cell. It's very naive. It's very primitive. And what we have seen in our Cellenkos, that we have a different pipeline, and FDA acknowledged that basically how we are defining our pipeline with phenotyping, even though I have to say that we are doing sometimes depletion, selection, and double selection. But again, the end product is with a different phenotyping of the cells. And basically the data, the clinical data that we have a positive efficacy signals. And and again, I cannot emphasize, during the COVID time, that was the only product was available and it could be shipped to us. And I think this is where the cord blood banking is allow us to work during the time that everyone was staying home and there was nothing out there for the patient in the ICUs. And this is where I think we have to think what else we can do with cord blood. I can not thank enough with the cord blood banking groups, and they are working with us. And we want to just, I guess, to emphasize there are areas that we can improve. One thing that, as a small company, we like to share that, at least the outcome of, like, for instance, there is no SAEs. None of our patients had SAEs for these products. And I think this is something that the future will help, that for other cord blood banks, that how to share data. I think this group can put something together. We need to just come to the table and just the area to help. Thanks to Colleen, Marcy, and Dr. Kurtzberg for allowing this to happen, and groups like us to come to surface and be here. Thank you.
Okay. Joe, you're on. Thank you so much for being here.
Thank you. Good morning, everyone. And thank you first to the conference organizers for inviting us to talk today. Thank you, everyone, for attending this talk. I'm here on behalf of RoosterBio, where I've been operating as the application scientist for about 7 years with this team now. Before that, I was focused in product and process development, where we're enabling the scalable manufacturing of regenerative cures. In this talk, I'll be talking about deriving MSCs from a perinatal tissue source of umbilical cords. So who we are at RoosterBio, we are a PD hub as well as a manufacturing capacity for research-grade materials. We have global footprint and presence where we support customers all over the globe. And we have manufactured materials supported by Type 2 master files as an ancillary material for further manufacturing purposes. We are hyper-focused on MSCs specifically because we believe that they are the building block to multiple regenerative drugs, therapeutics, as well as great production factories for secreted materials such as extracellular vesicles. Throughout the past 50 years, we've seen an increase or dramatic growth in the MSC space, whether we're looking at enrolled clinical trials, grants awardees, as well as different manufacturing developments and funding. When we compare traditional MSC trials to that of umbilical cord-derived MSC trials, we could see a large growth upwards of 2020 and a larger spike in the past couple years as we saw MSCs finding a purpose within COVID-related ARDS trials, which is not pictured here. We see that the number of patient enrollments has been increasing year by year, and we've also seen that the umbilical cord-derived MSCs have gained great popularity among these trials. When we work backwards or delve deeper into these trials, we are able to understand the manufacturing skills that are required for each of these trials, whether you're in a phase 1, 2, or phase 3 go-to-market commercial plan. When we're looking at cell therapy, a typical phase 1 is anywhere from 3 to 8 billion MSCs, whereas a commercial lot size may be upwards of 100 billion. When we look at EV therapies, these can range anywhere from 25 to a trillion, which is quite a higher number than the MSC standard dosage, which requires a larger lot scale or manufacturing platform to reach these targets. When we look at a conventional supply chain, or being able to derive MSCs or starting materials from a donor and bring them into a GMP facility, a typical timeline is anywhere from 7 to 11 years from standard research and development, upwards to product and process development, and then clinical manufacturing and tech transfer. What is somewhat of a bottleneck within the industry is that in between each of these phases, typically industrial customers have to go back to starting points or their starting point and reset their conventional supply chain timelines in between each of these phases as they're not built for scalable growth. When we're looking at research grade banks, or when they're developing banks for early research and development, a lot of individuals don't understand or don't think to the future of how would they scalably manufacture certain banks or starting materials to support large-scale manufacturing. This leads to a missed opportunity to start building in standardization and controlled practices within isolation, understanding desired CQAs or critical quality attributes, as well as understanding donor-to-donor differences early in your process to be able to ensure that you're bringing a scalable manufacturing practice online. When we're looking at what this means for cost of goods, this is extending your duration of development timelines, as well as inflating your cost of goods. So here at RoosterBio, we're really focused on standardizing the upstream supply chain of materials to enable the cost of goods decrease and the scale of manufacturing to be significantly increased. We do this by developing different manufacturing practices, as well as starting materials for individuals to leverage for off-the-shelf materials such as cell banks, EVs and exosomes, paired media systems like the expansion media, chemically defined collection medias for exosome production, and genetic engineering tools like genetic engineering medias to efficiently increase their overall transduction or transfection efficiency of these MSC cells. In late 2018 and early 2019, with MSCs being derived from umbilical cord tissue being a heavily patented space, we formed a strategic alliance with TRT Therapeutics, or out of University of Toronto, that patented a technology for isolation of MSCs from the perivascular region of perinatal tissue. This theory is that the MSCs are localized around the veins and arteries of the human umbilical cord, so we're able to start with a healthy C-section collected umbilical cord and derive the MSCs from this material. Our working structure looks like this. So we're able to derive a master cell bank at early passage, test for certain critical quality attributes before committing and bringing into a working cell bank at passage 3, relative population doubling level anywhere from 14 to 20 depending on the donor, and then we can leverage this off the shelf to support clients across the globe in specific manufacturing for their application. I apologize, there is a typo there with the P3, it should be P4. But typically customers leverage this material off the shelf and go directly into manufacturing, whether it's in 2D or in bioreactor platforms. These working cell banks are a model that we've created, and it is available from bone marrow, umbilical cord, or adipose-derived tissue, with the umbilical cord really offering significant advantages over the other two tissues. All of this material is available in research grade and GMP, which allows for mitigation of any bottlenecks as you look to transfer or translate into future clinical manufacturing. Lastly, this allows for that streamlined approach going from research grade to GMP and clinical manufacturing to enable a faster timeline into the clinic. Where our focus is bringing a high quality and standardized upstream material to accelerate the manufacturing of these MSCs or MSC-derived materials. By doing this, we're able to standardize the upstream isolation and the platforms to be leveraged, and then allow for individuals to start with qualified tissue before going into manufacturing practices. When we look at manufacturing within MSCs, typically this is in small-scale T-flasks or in multi-layer cell stacks, which does not offer efficient production of MSCs or yield efficiencies when you're in a scalable manufacturing practice. So in order to bring a feasible commercial therapeutic to the market, we theorize that your manufacturing practice must be sustainable, scalable, and then also enable a cost of goods reduction in order to bring a cost of goods feasible product to the market. And we've identified bioreactors such as the Sartorius STR Gen 3s, as well as the PBS Biotech bioreactors as efficient platforms for manufacturing MSCs at scale. In our initial process development, we were able to develop different critical process parameters that are specific for growing MSCs in suspension. When we're looking at here on the left, or on the right, is a small-scale PBS bioreactor, which is a vertical wheel bioreactor, and for lack of a better word, is a hamster wheel agitation technology, where we're able to isolate and understand different critical process parameters for expanding MSCs and eventually collecting EVs within these systems. From this baseline research, we're able to develop a process as well as bioreactor knowledge, which we can then refer to individuals when manufacturing MSCs at scale to enable their manufacturing or growth into bioreactor manufacturing at a quicker rate. We were able to look at multiple donors, and I want to direct your attention at the table, where we're able to look at multiple donors and isolate parameters that work across every donor, independent of inherent donor variability. In the top right, we're able to understand what a measure of success would be in terms of expansion, where the final density of any donor within these bioreactors was around 300 to 700,000 cells per milliliter. This process is typically a 4 to 5 day process, depending on the expansion time of the donor, as we do need to limit the expansion in bioreactors to ensure that there isn't too much aggregation and it would fault the downstream processing or achievement of a single cell suspension. So it is somewhat of a balance. What is critical is that when we compare this against 2D models or just standard planar culture, we're able to see that the MSCs are able to maintain all of their critical quality attributes, whether it's adherence to plastic, expansion post-bioreactor production, cell surface marker expression, trilineage differentiation into osteo, adipo, or chondrogenesis, immunomodulatory potential through the secretion of IDO1, or a relative angiogenic cytokine secretion panel. What is pictured here is all of the bone marrow data where we did our flagship development work with, but when we start to look at umbilical cords, we are able to see a dramatic increase in the overall confluency or achievable manufacturing scale. When we're looking at the yield, these donors range from anywhere from 700 to 1.25 million cells per milliliter, which is quite a bit, almost twofold increase in terms of production on a 5-day process as compared to their bone marrow competitors. We theorize that this is due to the relative smaller size of the perinatal-derived MSCs, as well as their relative growth rates in 2D as well as in suspension. An average population doubling time for the bone marrow platform is anywhere from 28 to plus or minus 4 hours, whereas with umbilical cord, we're around 18 plus or minus 4 hours across these donors. Thinking about this at scale, this offers a significant improvement in terms of manufacturability of these cells, as well as a significant reduction of cost of goods as individuals start to plan out their lot size manufacturing. Once again, in bioreactors, we are able to see the maintenance of all of the critical quality attributes of the umbilical cord-derived MSCs within these systems. This starts to help us develop a manufacturing plan or scale across different manufacturing systems. When we're looking at implementing these manufacturing protocols, we're able to support small-scale development work at research and development and predict where they would be if they were in a commercial scale or in a large-scale bioreactor at a phase 3 go-to-market scale. All of these materials really aid in the succinct processing of MSCs, as well as driving cell proliferation to meet clinical lot size manufacturing, whether they're in a phase 1, where we can see the estimated yield being 3 to 5 billion, depending on the bone marrow donor. If we're with an umbilical cord donor, this is anywhere from 5 to 10 billion, once again offer significant increases. When we're looking at EV manufacturing, this is once again the same platforms that can be leveraged, but instead of harvesting the cells, we can simply wash over to a chemically defined collection media for a purified collection material of EVs in secreted conditioned media. When we look at downstream processing for EVs, we can collect that conditioned media, clarify it through depth filtration or a filter set series, and then go into a very similar process to that of virus purification. The first step would be TFF1, where we're looking at buffer exchange and concentration of the material before going into guard filters and further clarification of the material, and then going into chromatography, where we can choose either anion exchange or size exclusion based approaches. This is removing any of the impurities from the process before going into tangential flow filtration 2, which is really lastly a buffer exchange and final formulation and storage. When we look at MySev criteria and look at different CQAs or critical quality attributes of the EVs from manufactured 2D systems or bioreactors, we're able to see relatively the same size and particle distribution, comparable mRNA profiles, comparable surface marker expression, as well as protein content, and then potency through simple wound closure assays. Lastly, at RoosterBio, since we've done all of the development work in terms of process design and manufacturing at different scales, we believe that without the hindrance of the transition from research grade to GMP, we're able to enable a faster timeline to scalable manufacturing, as well as generate a large amount of EVs or MSCs to support cell therapy and EV therapy manufacturing at quicker rates. This takes a long and windy road to a more succinct profile and a more streamlined approach into scalable manufacturing upwards to the 50-liter scales. Thank you.
Joe. Don't leave. Okay. Oh, look, Gazina's already getting up. You're not even going to let me ask my first question. She's like, I'm on it. We're going to invite all the speakers up, and I'll help moderate a session. Happy to take a question from Gazina, and then I'll ask some questions. And please, this is your opportunity to ask some representatives from companies who are using cord blood about their work and how we can better partner and collaborate. So.
First of all, I have a question to Joseph and then to Jesse. To Joseph, you would agree that the MSC derived from bone marrow or from the umbilical cord are different, right?
Yes.
But the pictures look similar. Do you had a chance to go with the cells into any kind of animal, classical animal model, mainly for bone or cartilage regeneration?
Yeah, so we are limited as a raw material supplier and process development hub, but we do have clients all over the world leveraging different systems. When working with these clients, we've noticed that depending on the indication, we have some clients preferring umbilical cord derived materials as some clients preferring bone marrow. A lot of tissue engineers really stick with bone marrow, I've noticed within my discussions, whereas umbilical cord, there's a lot of individuals looking at nuanced technologies or different platforms really focused on yield of MSCs because you're able to drive yield of umbilical cord MSCs to a higher confluency. So it allows them to reduce their cost of goods and manufacturing.
Yeah, and it's wise because the indications can be completely different for a bone marrow or a cord-derived MSCs. So.
Exactly, as well as donor differences.
Yeah, right, in addition. And then I have another question to Jessa, because I saw your expansion strategy. So you have, if you take cells from the segment, you have a mixture and a very minor percentage of CD34. So in the practical world, how long you expand them and how is your percentage then, or your purity of CD34 before you can go into NK cell development, because you need from the segment substantial numbers.
Right, that's a great question, and this is one of the things that our process development team is working on, where one of the amazing things we've seen, because we have this small molecule, that it allows us to keep the proportion of CD34s a lot higher, right? Without the molecule, they tend to all differentiate and crash out. And the other thing is the growth conditions that support those CD34 cells don't support a lot of the other cells that are in there to start off with. So there's actually a self-purification that happens over the course of the culture.
But how long is your culture period that you get a sufficient number, 90% purity of CD34 cells, for instance? How many days?
Well, we can actually get to, in the first couple of weeks, we get relatively high purity. But if we want to go a lot longer, we're working on, I think, some proprietary strategies for that at this point.
Okay.
Yeah.
Yes, okay, I'm going to try to focus in on some more basic questions, more around collaboration, networking, use of cord blood. So you guys all gave great talks, and you all mentioned why cord blood, but I'd like to ask, well, maybe you two first, and then I have a different question for you, Joe, to expand a little bit, no pun intended, on why cord blood. And did you start off knowing you wanted to use cord blood, or was this, did you look at other sources? What went into your final decision? I mean, I certainly know from our perspective, and I'd love to know what investors say to you guys too, or other people in the community, right? When we talk to investors, one of the things we say is cord blood is the only source of regulated starting material. And so we need to really hammer home the advantages. So I'm curious, what were your decision-making processes?
I think we have looked at other sources of CD34 cells, and from looking just at the micro level, just at the cells, cord blood CD34 cells are so happy to expand. They have to go, those are the cells that have to go and make a whole lifetime of immune cells. So you're able to put them in culture, and we've taken them out to 8 weeks, where they've continued to expand exponentially. Whereas if you take other sources, peripheral blood, mobilized, non-mobilized, we're still able to get expansion, and we're still able to use the molecule to get a lot more than we would otherwise, but you have really that inherent growth capability of cord blood is unmatched. And then there's, at the macro level, do we want to have a banked, regulated source, or do we want to be wrangling free-range humans to track down donors for bone marrow? The choice there is pretty obvious for us.
For most of us, yes. Although it's amazing how many people do not understand that concept. Tara, how about you guys?
First and most is available source. It's qualified, is ready to use. Also, the ones that actually the technology came from MD Anderson, they looked at the peripheral blood, and it's some mature cells, it's flip-flops, and the peripheral blood it becomes more effector cells. And mainly, I have to say, is the availability of the source was for this technology. It really made it really a no-brainer to use, and basically it's very naive and primitive cells that it's easy to work with. I understand a lot of people, they would look at it as like 2 tablespoons, how can you work with it and all that. But I think maybe 20 years ago, it was a challenge. Gamete cells came in, and I think after 20 years, there was a lot of bumps for them, but I think for us, for companies, small companies like us, it opens a different door. Again, all I can say is the source, and it's available. It's FDA has looked into it. It's licensed. It's tested. Like you said, that we don't have to chase donor, and then also the testing. It's a qualified product.
Yeah, so I think that's a big factor here, especially in therapeutics as opposed to generation of cell lines that can be made commercially available. Certainly, the infectious disease markers, I don't know, well, I guess you've been through the FDA, you guys are still pre-IND, but certainly I think that education around the fact that cord blood is viral free, the issues of infectious disease markers that we don't face with trying to harvest adult donors. Have you had issues with the FDA around infectious disease markers, or
no,
what has been?
The only thing was FDA, I think the first IND we had, they are very educated with cord.
I hope so.
They are. And then it was really a challenge in the old times. But right now, what they liked us to do, to use the US bank, and mainly, I was pushing because I'm coming from cord blood banking, and I wanted to go to Europe, other places. But I understand it. The testing, CLIA lab, it's more control. It's phase 1 study. Future is going to be different, and we provided everything that they wanted according to what basically guidance is. But I think here the standards for cord blood is very black and white, and we followed. But FDA was very comfortable that where the source are, and coming from accredited banks, licensed and registered, and basically we are using unlicensed product, but we can use the licensed product, but the cost is really high for us right now.
Yeah, it's interesting to me. I think, and anyone else who has comments around this, I think it's been fascinating to me that people don't understand that cord blood is a renewable source, even though babies are born every day. There's a lot of pushback that I've received when I've talked to, especially investors and general population. First of all, they don't know they call it cord blood. They don't know what cord blood is, and they think it's incredibly expensive.
It is, and they don't understand it's available. It's first and most, I think, it's education, and we have to, for cord blood, I understand, a lot of people, they understand the cell therapy, but when we talk with investors, we talk about facility or how to manufacture. They just shy away because it's too expensive. Most of us, we know that yes, it's expensive, but again, we can use the cord blood, expand it, have multi doses, and the cost would be much less at the end of the processing part of it. Yes, I think the R&D maybe it's not clear, but investors that we had, it took me 2 years, I think, and every time I was saying the same thing, very, very primitive language, and they were just coming back, what is that? What, CB, what? And it was tough, but I think Gamida, that got the license, they came back and they were just like, oh, now we understand you can license this product. So it made a big difference. And one of the other things is, I think, groups like here, that they can put, I think, more information out for a lot of people because they don't understand what we are in the cord blood banking. We're so busy. I think, I'm not right now doing the cord blood banking, but I feel like, it was just so busy. And that's where the education or, how we put the information out, it was not enough. I tried in my LinkedIn these days to put publication, put, what's T-Rex, because even with the products that we manufacture, there's a lot of, it's new, it's the past 4 or 5 years that the T-Rex is coming along for cord blood, but I'm so happy to see that the NKs, there are other products that is coming from cord blood. And I think as groups that we are working in cell therapy, we should educate. And we should, I guess, I agree with Colleen, follow the LinkedIn, post it. It's really important that, everyone, the audience, they understand what the cord blood is. We think everyone knows, but truly, we are a small group.
It's not just for transplant.
No.
Joe, so you talked about a proprietary method that you guys have in your hands. I'm curious if you could speak to how you do source your cord blood, or your cord tissue. And have you, because you can make massive scales in working cell banks and master cell banks, are you working with the banks to acquire this, or are you doing one-offs on your own where you're saying, I'm going to work with hospital X and I'm going to get the, you said from C-sections. So how do you source your material? And since you've got a lot of bankers in the room, would you work with banks versus doing it on your own if you're doing it on your own?
Yeah, luckily we do have a strategic partnership with a qualified vendor. So the work to put together the viral screening as well as the collection period as we are processing fresh tissue was a very large undertaking, especially during COVID era, when the first banks were derived for GMP. It was very difficult to do, but luckily we had a commercial vendor who we were very close with from our partnerships, where we were able to lay out a set of terms and criteria for the cord, and then look for the correct donor.
So when you say vendor, that's not a bank?
Somewhat, yeah, they are a bank.
Okay. Yeah. No, I'm just trying to, and certainly
Yeah.
that's not I'm
Yeah.
just, curious and to try to open up the discussion, because I actually think you bring up a really important point that our cord blood banking partners provide, right? It's that qualified material, already screened, right? So all of those things are incredibly important when you're bringing in your source material to then make it therapeutic, right?
Definitely. And being a global company, we know that the APAC region is very, I would say, preference towards umbilical cord derived MSCs. So thinking about global compliance, some of the requirements from tissue banking here in the United States may not be or as stringent as outside the United States. So that's something that we had to take account for and work with individuals and kind of customize our collection plan, right? It wasn't something that was already built. It was something that we had to work strategically with an individual.
Okay, I'm going to ask one more question, then I'll open it up again. But I'm curious if you could each speak, Jesse, I'll start with you. to what your bottlenecks are right now in getting, because again, this is gonna, again, please, and other folks here who have other representatives from other companies, feel free to jump in as well. But what are your bottlenecks in getting your source material? What are the big hurdles that you face?
For us,
For research and thinking ahead for clinical.
right, well, exactly. So we have a large supply of segments. We've worked with the New York Blood Center to get some segments, and we've had the ready availability of ones that we can use to demonstrate in our research to show donor variability, to show differences in potency, all of that. What is a bottleneck, and actually part of the reason that we've come here is to have the ability to have a large number of segments come through the door so that we can do this screening. Because when we were talking to the New York Blood Center, he said, okay, well, we want to buy a segment. I said, well, no one's ever asked to just buy a segment before. Like, we don't know what that is. We have to go back and we have to have conversations. And so this is a new conversation that we're having every time, is what does it take to get the segments? And the segments of not just research cords, but the segments of cords that were we to say, hey, oh my gosh, this is an amazing donor, we want to take this through to clinic, that we can do that already. So that's really the main one for us.
And a lot of that is just for validation purposes, right? To do your research and be able to initially show what you're saying, the donor-to-donor variability, just so you don't need the whole cord.
Well, the idea is to have the segments to show the variability and then to actually identify the ones that are the top performers and then go back and get that cord. So it's really about being able to have the full access.
So if a bank maybe had a bunch of segments lying around and no cord attached, would that be helpful for you guys in any way initially, or to do some of this? I know you wouldn't be able to go back and get the cord, but would something like that be helpul?
Well, that's what we have.
I don't know if
an
banks have
arrangement.
that.
We do actually have that right now. We have
So that's
we started with that, but it's really that taking it to that next step.
But does it have to be segment? Cannot be the cord?
Well, the idea is that we want to non-destructively be able to assay the cord. And so it could be a tiny amount of a cord, but we need really 50 microliters of cord blood, right? 50 to 100 microliters, that's enough to turn into a full cord's worth of CD34 cells that we can then run through all the NK differentiation, all of the animal models that we need. And so it's actually very wasteful even to use that 20, the ones that we get are 80-20 bags. Even to use that 20% is wasteful because of the expansion. So we wind up cryopreserving a lot of that along the way in the research process.
I guess the challenge is that, when you have a segment, 3 segments, and usually for transplant we use one of them for confirmatory testing, then we have 2. And I know even me getting the cord, I usually ask for having the segments. And I guess that, that's something that, with our banks, we said that just like, they gave me 1 segment. It's just like, no, I want all the segments because, and then that would be, I think, a loss of the cord usage for the bank. And I don't know how that can be.
So that's why we're looking to try to get away from the ones that are likely to be actually going for transplant. So move into that range where they're the more common HLA, the lower TNC.
I love the idea of screening, though, right? To not use the unit that you don't
true.
need. Joanne?
I just want to say as banker that, and I think, Jesse, your idea of dividing the inventory into buckets of, cell doses that are likely to be useful for transplant versus other. And in a bank like ours, where we've been banking a long time, and I know this is true of others, our early inventory, which is still very stable, you saw that with Gazina's presentation and Jim's presentation, we have smaller aliquots of cord blood that are very unlikely to be used for transplant. We know the HLA type, so we could even select for common HLA if we wanted to. And we can give you segments from those units without any worry about not being able to provide them for transplant. We have 3 segments, so even if we gave you one and then we needed another one because a patient suddenly appeared that needed that unit, we could work with 2 segments for that patient. So I think, I'm saying for our bank, but I think it's true for all the banks, that we can make buckets of large numbers of units that could be screened.
That would be amazing, yeah. We really look forward to having a conversation about that.
Any hurdles or bottlenecks that you want to talk
For
about?
us, I think it was just like the cost, because we want juicier cord and we wanted large cord, and sometimes I think it's, and we are working with the banks. I think they were generous with us, but it's obvious if you have a bigger cord, the TNC, you can have more doses, but the cost, it goes really high. And at this stage for us is really kind of a challenge. And also, I think we had earlier on more challenges. It's getting better every year because we understand what the question we should ask and how they communicate with us. But I think it's basically the cost, and that's the only thing I can say.
So the cost per unit,
Yes.
right, as your starting
Yes.
material, right. Yeah, I agree with Joanne. Jesse, you had a nice slide, and I think a lot of us in the cell therapy space do not want to touch that upper right quadrant, right? We've seen testimonies from patients here throughout the conference, and the intent is certainly not to deplete transplantable units. The intent is to help the banks mobilize inventory that wouldn't necessarily be used for transplant. So I'm just going to pause for a minute. Is anyone going to ask any questions? Marcy?
Thank you all for your presentations, all really excellent. I guess I have a comment and a question for Jesse. So one comment is I think that your approach about segmenting that inventory and then thinking about prospectively making the kinds of units or products that you want is really, it sounds really unique, but I want us all to think about, most of us do that with HRSA. That's exactly what our HRSA contract is. So I think we have experience doing that, but we haven't had people come to us thinking about proactively developing the products that you want. I think the banks all have that capability, but no one's really asked us in such plain language, what if we wanted X number of units? We do it every year with HRSA. We negotiate a contract with them. They say we want units have to be 0.9 or better and all of these attributes, and we have the whole year to deliver them. So I think we should all start thinking about we've already done it, but let's think about how we could do it for you. So thank you for bringing it up. My question to you is, I think you've done a great job of showing how you use these segments to determine the units that functionally work for you well. Have you looked back to see anything about the donor itself? Like if we could predict what donors, before you even look at a segment, might make a good product for you?
Right, that's part of the model that we're building as we go through and accumulate. At this point, we've run, I think, a little over 100 segments, and so we're starting to build some of the associations there. Yeah, this is. I mean, it's going to be everything from looking at genetic markers, seeing if there are parameters of the HSC expansion. What are we seeing that tells us all the way back to, what's the sex of the donor? Is there an HLA association? Is KIR-B really the be-all, end-all? So these are things that we're looking at in building this comprehensive model.
Any other? I would love to think about the banking part of things and that inventory. So Marcy, you just brought up the HRSA contracts you guys have, and I know we've talked about this a little bit, but part of the problem is how do we and other folks in here using cord blood as source material, how do we access those units that have been subsidized by HRSA or contracted by HRSA when those units, as I understand it, I'm not a banker, but as I understand it, those, if you give them to us, you have to replace them, and they are supposed to be patients' specifics. I see a lot of nodding of heads. So I'm curious, because really what I would selfishly like to see, right, is somehow to access inventory from all the banks that you just are never going to use for transplant. So, but instead of trying to work individually, right, is there just a pool of these units that you know we're never going to use them for transplant? So how do we all, as companies developing therapies, access those without hurting you guys? We want to lift you guys up.
Well, I think there's a time frame too, right? There's a process. So I think there's a way to keep them available to everybody, and then if it gets pulled for a patient, we know there's a CT that has to happen, so that takes time. So I think we all could segment our inventories based on the likelihood of them getting used for transplant. I think most of us already do. So I think there are some buckets that we wouldn't make available for this kind of technology, but there's lots of buckets that we would. Because the likelihood of using them is low, we probably have duplicate ones in our inventory. And I think the HRSA question is a good one. Right now, you have to replace units that you use if it's not for a designated patient under an IND or BLA. But I think there's room there. So if we could also recover our reimbursement from HRSA, we would probably use it. So I think there's a balance, and we have to start thinking about it in a different way, too. And continue to have conversations with HRSA, which we are having. Joanne's organized an all-bank meeting, and we talk about these alternative uses every time with them. So I'm sure you.
And I just want to say, I think HRSA, if HRSA can hear that, not only from us, because we're already telling them, but hear from groups like you all, that this would be very valuable to developing products that will help many patients. I think they're open to thinking about modifying the current rule. In their defense, their contracting systems are very cumbersome, and changing old contracts is harder than prospectively changing new contracts. So there's all that, but I think HRSA is listening, and maybe even in the next advisory council meeting, if an industry group or coalition can come and make a short presentation, that would make a difference. Yeah.
I'm sure any one of us would like to come, or we can come en masse.
One of the things that, I know we approach as a company to a couple of banks that we would help to help with the infrastructure for the collection. And, the courts, because like for our purpose, we don't have to use the ethnicities that they are really important for the transplant. We use majority what you all don't want, you give it to us. But we can partner that we pay, and then, it stays, the bank collects it under their standards, but it would be like if there is a patient that comes and the HLA matches, it would go to a patient. But if it doesn't, then we can get it, because this is at least helps the bank to not basically think, where am I going to get the money? Yes, HRSA is going to give the money, but it's just for recovery and this, sort of thing. But at least it helps, like, for FTE, helps with the regulatory. I think that's one thing. The companies, I think, can, like us, we can partner with the banks and help at least for the collection. We don't need to have the court; it would stay there. But when we need it, they would just the costing that we can get it.
So in terms of like Marcy's suggestion of prospective collections with some of us around, still maintaining the highest for you and the rest for others, especially if you don't need HLA.
And it would be available for the patient anyway.
Right, no one was, And I agree with you. I think HRSA would be very open to life-saving therapies. Who's not, right? Leland?
Well, great talk. I just have a question in terms of not necessarily cord blood, but cord tissue. And this is more of a bank question, but I'd be interested to hear what people have to say. What are the difficulties compared to cord blood banking in getting cord tissue and storing it, validating whatever process needs to be validated, and what you utilize it for, and is it,
I can't answer. I don't collect cord tissue, what bank, I'm sure there are many people who are collecting cord tissue. That's what you're asking, Leland, right? Is it hard? I'm like, why aren't all the banks cord blood banks collecting the blood and the tissue?
Yeah, like what?
Like, why not?
Yeah. So.
I mean, Joe, you might, well, do you know? Or is that more of a?
No, I think this is a really important issue for a lot of reasons. I think cord blood banks easily can put SOPs in place to collect cord tissue from moms who are donating their baby's cord blood. And I mean, just as an example, we do that, but we do it from C-sections so we can keep the tissue sterile without adding antibiotics. But there are a lot of approaches, and cord blood banks know how to be GMP and know how to put the right SOPs in place. I think coming back to the purpose of the CBA, I think one elephant in the room is that a lot of private banks are storing cord tissue in a variety of different formats. And I don't know that all of them have demonstrated that cord tissue can be processed at a later time to produce real therapeutic cells. And I think the whole issue of MSCs, for example, from cord tissue is really, really important to think about. How do you manufacture? I mean, we've heard, but how do you scale up? Is that a personalized product, or should that be really a third-party manufactured product? In my opinion, just my opinion, I don't think you can put a chunk of tissue in the freezer and come back later and make a large dose of viable MSCs, for example. But I think if you start with the fresh tissue and do some early processing to get to P0, for example, or P1 even, then you've got something to work with for later expansion and product development. But I think in the field of cord blood banking, when you think about what's happening on the private side, what's happening on the public side, or the family side, and the public side, it's a real issue we haven't addressed yet. But definitely, back to Leland's question, public banks have SOPs to procure the tissue. And again, in our case, I would say you have to do some early manufacturing before you have material that you can use for large-scale manufacturing.
And that definitely is something that was a challenge, right? Is the collection of fresh tissue, your shortened window to start the processing,
right,
while ensuring that it's sterile, already past the viral screening, right? So there's a lot of risk associated with that process in the first 72 hours, let's say, as opposed to bone marrow, where it's a little bit more flexibility in the pre-screening process. And you may have a higher success rate under GMP manufacturing with bone marrow or adipose, where you can do a lot of the testing up front, whereas in cord tissue, there's some risks involved in starting the process early on the fresh tissue, ensuring that the surgery went as planned, right? Or if you ran into any issues with some of the screening there.
Yeah, I mean, I think you can do it easily with the cord tissue also, because the early expansion doesn't take a lot of resources.
Yep. Yeah, definitely. What we've run into is that, with the very stringent, I would say, viral panel, as well as the consent forms, we touched on it, I think, at one of the talks yesterday. We found with RUO cords, and for people donating for research use, there was a higher volunteer rate, let's say, to give away the cord for that, whereas with the GMP for further manufacturing and commercial use, some hesitancy from different patients.
Again, and I shouldn't be the only bank talking about this, but I think some of it is how you present it and how you explain it. But I do agree you have to have consent to do that and to be making third-party products. But I think enough donors are willing to allow that to happen.
Yep.
Are there other, I have one more, well, I actually have a lot more questions, but are there other questions from, and we only have a minute. I want to make sure I want to allow you guys access to these guys. Asako?
Hi, I don't know who to ask these questions, but it's related to the, what Jesse was talking about. But we see lots of this donor variability in the cord blood, and then that's like one of the biggest challenge that we have to use when we use cord blood for the industry purpose. So Jesse is I guess selecting the cord blood that's suitable for the NK, right? And then we are doing a CD34 expansion, and then we see lots of donor variability, and it's really very, very difficult. So I'm wondering, well, I guess that this donor variability is true probably for the transplantation as well. So I'm thinking, there is any possibility for, all of the people here for the, banker to collaborate and then figure out and, get to the bottom of these issues of donor variability. And then, I know it's a very, very difficult $1 million questions, but if we can kind of get the answer for this, and then I think it's gonna really help to move the industry. So I don't know who to ask this question.
And just as a quick answer, one of the things that we're looking for in building this model is not just to understand what makes a good cord for our manufacturing process, but if there is data on, or if we can create data on markers or profiles of the cells at time 0 that will tell us that they're good expanders, and hopefully that also can go back to the cord blood banks and look at how well that qualifies a unit to be successful in transplant even, right? Like hoping that creating more information around donor variability will benefit everybody.
Yeah, and I will say donor-to-donor variability is a huge issue, and that's why we do pooled donor manufacturing to get over that. But I will say as a transplant physician, though, the one downside of that is we don't want to limit the available donors, right, when we don't really understand, because graft failure is not really a problem anymore in cord blood transplant. So time to engraftment varies, right, just like donor-donor variability. But my fear is that we're going to get information that we don't know what to do with, and then all of a sudden start knocking people off the donor registry. We used to collect, just a funny story, we used to collect cord blood, what we'd still do for research at the Fred Hutch at a local hospital, and that's where I delivered my son. And my notice to the lab that I had delivered was that his cord blood was collected and sent to the lab for research, but no one would use it because they were afraid if those cells didn't expand, that somehow I was going to be worried about my son for the rest of his life. So that, I think, is still somewhere in the freezer at the Hutch. But yeah, so I think it's a sticky wicket. We're out of time, but I know that everyone here and the bankers and other, technicians and everyone, please come up and ask questions. We really want to facilitate networking and collaboration. And this is really what the association is supposed to help facilitate. So we have a break now, and then please come back for the last session.
Yep. Thank you, everyone.
Thank you.